Hello everyone, this is Dr. Sachinanand Dotto, and in today's lecture in marine ecology series, I'll talk about on environmental DNA. So environmental DNA is totally a new approach and a new tool to detect the species in a particular ecosystem. So let's see about the environmental DNA. It's a very interesting topic. So first of all, we have to know what is environmental DNA. Environmental DNA, or we can we can call it's a eDNA. So it's a technique that we are collecting the sample from the ecosystem or that particular habitat without capturing the species, and from the environmental sample, and we detect the DNA, then identify the species, and that sample are coming from that environment or the ecosystem, and that species excretory product, or it could be feces, or it could be the genetical product of that species that are released in that ecosystem. So we are not capturing the species, the samples are collect from the environment, then detect the DNA, and then identify the species. And as we all know that all the living organisms have the DNA, and DNA is a main genetical material of the any organisms, and each and every individual in the planet Earth have a specific or unique DNA. So DNA is a very important tool to identify any individual of this planet Earth. So each and everybody have a separate DNA. So it's a very important tool or important technique to identify the species, and the accuracy in this case is very high, and it is also very cost effective. So eDNA, basically DNA released from an organism into the environment. Okay, so eDNA basically it could be the samples or the DNA that's coming from the genetic material, or directly or indirectly it's released from the environmental samples. That are the sample, maybe a soil sample, maybe a sediment sample, maybe a water sample, without any obvious sign of biological sources materials. Okay, so no need to capture that particular species, and no need to take the sample from that particular individual or particular organism, like the blood sample or the tissue sample. So whatever the floating or available sample available in that particular habitat, we are taking that sample from the environment and that detect the DNA. So that samples may be coming from the skin, hair, scale, intestinal tract, maybe the germ cell, or the just sperm and eggs, and it can be the remnant from that particular species or individual. And so eDNA is basically a relatively new monitoring tool, and its featuring is a very increasing number of application. We will check the application of the eDNA at the end of this lecture, and it's also facilitates of an accuracy. So I have talked about it. So DNA, each and every individual have a unique DNA, so its an accuracy is very high to detect the species, and very cost effective, and also the you can identify the species from the environmental sample. And for the higher organism, DNA may come from the excretory cell, the tissues as like urine or hair or the skin, or a dead individual. You can lacking genetical material could be a sample. Okay, and free floating DNA means from the aquatic body you can find all the organism are releasing basically germ cells, releasing the genetical sperm and ovum, and also the urine, and also the excretory product. It can be used as a sample, and you can also take the microscopic taxa like plankton. Okay, and the environmental DNA methods is basically totally new approach for monitoring the species distributions and abundance. So abundance is not that much appropriate, but it's you can estimate the abundance. Mainly is the identification of the species and their distribution of that particular habitat. So in this method, DNA based species identification or DNA barcoding, so each and every individual or the species have their DNA barcoding, and so that sample, whatever we have the database. So from after extracting the sample, we are also match with the database and then identify the species. And so DNA barcoding that is used in order to detect the species through the extracellular cell. Extracellular cell means that cell is not taking from that particular individual or organism. It's not coming from that extraction of the blood or extraction of any cell or tissue from that organism. It's the coming from the environment. Okay, and that is called basically cellular debris and present in the environment sample. And and the relationship between the eDNA concentration of the species and the abundance by biomass have previously already means whatever already reported. So eDNA concentration in water sample is an example I'm giving here. The water sample, eDNA sample, it's maybe the similar species abundance index, and the invasive capture method that is basically used for the fisheries management for fish species. So it's a very important tool for the management of the natural resources or biological resources, and also making the policy and the recommendation for the management of the species. Okay, and also the habitat. Then the habitat of the eDNA. So what are the possible habitat for the eDNA? So it could be a aquatic habitat, it could be a terrestrial habitat, or it could be a animal traces. So aquatic habitat, the lentic, lotic, and the marine. Lentic means it's a stagnant water, ponds and lakes, and geolotic means flowing or floating water, that is river, stream, creek and spring, and the marine environment. Okay, and terrestrial habitat, we are using the soil as a soil sediment as a sample, and the ice core could also a good sample. And the animal traces, it could be the feces, it could be the urine, it could be their track or pathway, and it could be the hair, it could be the different means excretory product or sample from the animal. Okay, so these are the possible habitat that we can extract the eDNA. Then the technique, so it's a very important that how we can estimate the DNA from the environmental sample. So for the aquatic species as example, so eDNA basically captured by filtering a large volume of water. That why large volume of water I will explain later. Okay, so basically it's required large volume of water, and after getting taking the samples, you have to make it concentrated, and after the concentration you have to make a PCR. But that PCR means whatever sample you are getting the biological sample, then you have to do its PCR. PCR basically polymerase chain reaction, it's basically amplify the DNA. So it's a good technique that can be, if you have a very low sample, low DNA sample, so but due to using PCR you can amplify your low sample into the high sample. Means amplification is very important for targeted region of DNA, means specific region. So that is called metabarcode analysis, and making it possible to detect the quantify the species specific DNA sequence, and that is basically taxonomically assignment. So as I have already talked that species specific DNA is each and every individual or each and every species have their unique DNA. Okay, and that PCR analysis basically used for amplification of mitochondrial gene, and it could be the species specific DNA primer. So basically the primer is a specific primer or universal primer. It's already established and already have the particular sequence of that particular species, and that primer can be used to identify the species. And so we'll see the sampling technique here. I have talked about that why we need to lots of the water sample if you sampling in a aquatic body. Why? Because if you sample in a particular region or very limited water for your sample, then you cannot the species cannot be detected, because in this sample it could not be the species means other excretory product or any sample or the DNA sample could not be present. But if you sampling from a vast or a here and there randomly, or you are taking a lot of sample, means a huge water sample, then it's possible that you can detect the species. Okay, So that's why sampling is require a huge amount. So here flow diagram of the full technique. So first of all you have to take the water sample, then you have to concentrate the sample, means making the huge amount sample into a very low concentrate, means low sample amount. So by concentrating it, then you have to extract the DNA, and then after extracting the DNA you have to amplify it through the PCR, and also using the universal barcode primer. So universal barcode primer is already established, or it's a particular same sequence, a small base pair sequence that is also available. Okay, and then after doing this PCR you have to making the sequencing, so next generation sequencing, and after that you have to compare whatever your reference database. So each and every species already database is there, then you have to compare with your sample with your reference database, then you have to identify the species. Okay, then you can identify the species. So it's basically a meta barcoding technique that using the universal primer. Okay, so in ideal universal primer for the species of DNA, and basically it's amplify very short fragment that only 200, less than 200 base pair only, so ATGC 200 base pair, and it contain sufficient sequence variation incorrectly assign organism to taxonomic family, genus and species level. So you can identify from this sample to match with your reference database, and you can identify at the species level, genus level, or also in family level. Okay, and it is basically useful, very useful for taxonomically and also the ecologically, because you are not capturing the species ecologically diverse range, because in the ecosystem also there will be huge diversity of the species. So it's versatile to collect it in terms of ecologically or habitat wise. Then along with the means DNA sample link, or you have to also taking care of the physicochemical parameter of the aquatic bodies, if you are taking from sample from the aquatic bodies or water sample, because it is very important to identify the species. Not only that, along with the habitat, along with the different physical chemical parameter, it is very important. So that's why analysis of physical chemical parameter, you need to check different physicochemical parameter like the temperature, pH, transparency, color, velocity, turbidity, DO, total alkalinity, hardness, and these other parameter, and also along with the biological parameter like the zooplankton, so phytoplankton and fish. So that is about the whole complete study of the DNA, environmental DNA. So advantage, now talk about the advantage. So it's a very sensitive, very sensitive. So why? Because of the due to the higher chance of detection. So as it is very sensitive, and so detection of the species is a very high chances of probability, and especially for the secretive species, I mean which species of secretion you can get. Okay, and or the species occurring at the low density also you can identify, and less effort is needed to detect the species using the eDNA method when comparing the traditional sampling technique. So traditionally, if you want to sample link different biological, I mean biodiversity methods, if you do the quadrate or the sampling different line density or different sampling techniques, much more effort full. Okay, in compared to eDNA is you take the water sample and analyze it. Okay, so it's a very cost effective also. So it's very cost effective also. So you can identify many species for a one sample. Okay, in compared to the traditional methods. Okay, it is also very cost effective, and it's much more environmental friendly. Why much more environmental friendly? Because you no need to capture the species, you no need to disturb the habitat, you no need to means damage the habitat, and also no need to detect catch the species for their presence, and also no need to take the direct sample from that particular individual. Okay, And so high specificity is also, so misidentification can also be excluded, because here the identification is under almost 100% accuracy, or maybe 90 to 95% accuracy. Okay, so validity of species specific, so because you are using a species specific primer. And finally, the eDNA methods can, in some cases, provided higher taxonomic regulations is very high, and especially in case where the species cannot be distinguished based on the morphological character. Sometimes morphological character is very difficult to identify species from the morphological character. So in that case, eDNA is very important or very useful or helpful. And disadvantage, so each and every technique there will be advantage, disadvantage. Here the eDNA is also few disadvantage, and most of the disadvantage I'm counting here from the marine environment. Okay, so because in case of marine environment, it's a large ecosystem and large area, so you need a very high amount of water sample. Okay, and also the effect of sea current, or the waves, or the winds, or the waves, or high tide or low tide and waves, this basically disrupting or the dispersion or dilution of the eDNA, means it can be bring the sample from here and there, so it's a problem for the marine environment. And salinity is also impacting the for the extraction of the eDNA from the, whenever the biological sample are released in the environment or in the aquatic body, in the marine environment or marine water, the salinity is also impacting on the extraction of the eDNA. And positive detection of eDNA only possible if targeted species in area recently visited, means if because if time, or means few time after or you go, means the time is gone, then the DNA can be degraded. So DNA can be degraded over time, so you have to take the sample, I mean, if the species is coming on that region very recently. So, and the negative result does not imply, so if you got a negative result, mean there will be no samples, is there no DNA mean sample you are getting, so that does not mean that there is no species are there or no individuals are there. Okay, so it could be a misinterpretation. And no quantification of number, so it's difficult to quantify the number of the organism in the habitat, so abundance is a little bit difficult to estimate. Okay, and cannot give a real-time information on the organism locations, because you are getting the sample here, the species can be moved from here to here, so regions means temporal or means spatial information of about the species is spatiotemporal information about the species is a little bit difficult. And possible expansive, it could be expansive because if your sample have lots of the eDNA, lots of the DNA is there if in your sample, so it's all the species you have to, I mean, identify with your record, so it's a little bit expansive on that case. Now application, so I have talked about that it has a huge application, so we see what are the applications. So it can be reduce the field survey time. So if you go for the manual field survey or the traditional field survey to identify any species or detection of the species and their habitat, so that is very means it's a time taking, but EDN is very short, it's a field survey time, no need. Okay, and only just take the sample from the environment. Okay, and have a little or no impact on the ecosystem. This is very much important, that eDNA does not have any impact on the ecosystem, because you are not disturbing the habitat, you are not disturbing the organism also. Okay, and species identification from the DNA sequence is often easier and accurate. Okay, I mean accurate again and again. Okay, then identification by observation of external morphology. So morphological characters somehow difficult to identify. Okay, and in that case it's a very useful technique. And varieties of aquatic species may detect from a single water sample. You can very many from one sample you can detect a varieties type of species. And eDNA collection can successfully be monitored. You can monitor your sample and you can monitor the whole thing. Presence or absence of rare, endangered, indicator, invasives or key species. It's very important from your sample that if you getting the rare endangered species, and for the assessment of biodiversity of the particular region, and detecting rare cryptic and elusive species, also detecting migration behavior, spawning behavior of the organism, because if you get the sperm or ovum in a particular time or particular region, then you can identify the species are being this is their breeding count, or you can identify their migration and spawning behavior. And monitoring species abundance changes in time, so abundance you can estimate over the time. And determine the species assemblage. So in a particular region, if you in a sample, if you are getting lots of the species, then you can identify, you can tell that the assemblage, species assemblage, what are the species are happening on that coming in that particular region. So assemblage is also important to identify, can be from the eDNA. And also evaluate management action. You can like fisheries management, like habitat management, habitat restoration, like the species restoration or invasive species control. So these are the different management technique you can take from the eDNA. And archive records, so it's very important. You are making a huge database for the future. And feature direction, so eDNA approach can also fully mature and integration into the conservation. So for the conservation, eDNA is very much important. Okay. And the temporal and spatial distribution of the species from the eDNA, various environment provide information about the what proportion of biodiversity is observed in time and space. Okay, so by using the eDNA in future, you can predict the observation of that particular species is present in that time and space. And more specifically, whether this is measured as a total biodiversity or total biomass of an individual density in relation to eDNA concentration. If the eDNA concentration is very high, then you can estimate the total the density and the biomass also. Okay. And abundance also. And the extract origin of eDNA are epithelial, mast cell, intestinal cell, feces, urine, and maybe the in future it could be the sampling, maybe it's increase and differ in the lifestyle, and thus it can be help in huge effect on the abundance figure. So abundance can be detect in future also. And physiochemical parameter, I have already talked about it, that not only the DNA sample, you also estimate the different physiochemical parameter for that particular habitat, like the temperature, salinity, pH. That also help you to understand the species relationship with the environment, and also the species habitat, a clear picture about the total ecosystem. So that's all about it. Thank you for understanding of eDNA. It's an interesting topic, and if you have any query, just comment. Thank you. Bye.